in the quest to understand the complexities of life and the universe. Groundbreaking research by Levin et al. Has opened new avenues in the study of bioelectric phenomena and their profound implications on biological regeneration. This research, coupled with innovative experimental proposals within the Genesis framework, offers a glimpse into the potential manipulation and understanding of higher dimensional space memory and the preservation of bioelectric anomalies. This blog delves into the significance of these studies and why they are pivotal in advancing our knowledge of biological and cosmological organization. The research led by Levin and his team explores the fascinating world of planarian flatworms, known for their remarkable regenerative capabilities. Unlike most organisms, planarians can regenerate entire bodies from small tissue pieces, challenging our conventional understanding of biological development and form maintenance. Levin's study particularly focuses on how bioelectric patterns, rather than genetic codes alone, dictate the complex morphological outcomes during regeneration. This discovery not only shifts the paradigm in developmental biology, but also aligns with broader theories in theoretical physics and cosmology, suggesting a deeper, more intricate connection between life forms and the fabric of the universe. Building on Levin's insights, the Genesis framework proposes further experimentation to explore the entropic dynamics within higher dimensional spaces that influence bioelectric phenomena. The framework hypothesizes that life itself may be a manifestation of higher dimensional entropic anomalies, regions in space where entropy or disorder deviates from expected patterns, leading to the emergence of complex, ordered systems like living organisms. The experimental proposals aim to manipulate and measure bioelectric fields in planarians to induce and then revert evolutionary ancient head forms. By doing so, researchers hope to observe the speed and manner in which these induced forms regress to genetically supported structures under different electromagnetic conditions. These experiments are crucial for several reasons. Understanding dimensional interactions. They could provide empirical evidence supporting the concept that higher dimensional space memory influences biological processes on Earth, bridging gaps between quantum mechanics, general relativity, and biological sciences. Preservation of bioelectric anomalies. By altering and then attempting to stabilize these anomalies through external controls, scientists can better understand how environmental factors affect biological aging and regenerative processes. Innovative medical applications. Insights gained could lead to revolutionary approaches in regenerative medicine, potentially offering new methods to control and direct tissue growth and repair. At a fundamental level, these experiments challenge and expand our understanding of the universe's operating principles. They suggest that what we observe as life and consciousness might be deeply intertwined with the cosmic fabric, governed by rules that transcend our traditional three-dimensional perspective. By exploring these possibilities, we not only unravel the mysteries of biological existence, but also enhance our understanding of the universe's structure and its evolutionary history. The integration of Levin's findings with the Genesis framework's experimental proposals represents an exciting frontier in science that merges biology with cosmological theories. As we continue to test and expand these ideas, we may find that our place in the cosmos is even more integral and interconnected than previously thought. These studies not only illuminate the path to understanding life's complexities, but also how we might effectively harness this knowledge to influence life's physical and biological outcomes. 1. Head regression speed in a Faraday cage. Objective: to test if shielding planarians from external entropic forces influences the rate or pattern of head regression. This experiment could reveal environmental forces that contribute to the aging of bioelectric potentials and morphological changes. Method. Place. Planarians with an altered head shape that deviates from their genetic coding in a Faraday cage to block and control external electromagnetic influences, and observe if there is a difference in the speed of head morphology regression compared to a control group outside the cage. 2. Effect of natural Earth frequencies on morphological stability. Objective: to explore if exposure to natural electromagnetic frequencies like the Schumann resonance at 7.83 Hz affects the stability or regression of head-type morphologies in the planarians. Method: expose different groups of planarians to various controlled frequencies within a Faraday cage and monitor changes in head types and regression rates. Compare these results with those from planarians exposed to artificial or higher frequencies to assess if natural frequencies support a more stable or slower bioelectric aging process. As shown by the speed of regression from ancestral morphological traits induced by bioelectricity to local 3D spatial backup encoded within DNA-supported traits. 3. Long-term observations of bioelectric modifications. Objective: to understand how long-term alterations in bioelectric fields impact the bioelectric aging process in planarians, specifically whether sustained bioelectric changes can permanently alter morphological outcomes if entropic aging can be slowed down. Method: apply continuous bioelectric modifications to planarians and observe the long-term effects on morphology. Assess whether these changes are reversible and how they correlate with the planarian's genetic default settings. 4. Testing lineage. Jump stability in bioelectrically altered heads. Objective: determine if bioelectrically induced head types that regress to evolutionary ancient forms can be stabilized under specific environmental conditions or if they inevitably revert to the genetically supported form. Method: utilize bioelectric field manipulation to induce ancient head types and then use a combination of environmental controls, including EMF shielding and specific frequency exposures, to attempt to stabilize these forms over extended periods. These experiments aim to bridge the gap between theoretical predictions of the Genesis framework and empirical biological research. By focusing on the interaction between bioelectric fields, DNA, and environmental factors, these studies could significantly advance our understanding of how higher dimensional influences affect biological aging and regeneration, potentially offering new insights into the fundamental nature of life and the universe.